very short, right? So let us talk about what is DNA fingerprinting. So basically, let us open our file for that. So DNA fingerprinting is basically your fingerprints, right? It has a unique property and it is different from every human being, right? So its basic theory here, nothing new about it. It is known as the it is known as that every individual has unique fingerprint, right?
Yes, sir.
These occur at the tips of fingers and have been used for identification for a long period of time. With the help of fingerprint, we can identify a person, right? A sequence
Yes,
of
sir.
bases of DNA is also unique for a person, and info regarding this is called DNA fingerprint. If we are looking at the fingerprint, they possess a specific type of DNA arrangement, a basis of DNA arrangement. So that's why they have specific, uh, what you will say, identity, right?
Yes, sir.
It is same for every cell and cannot be altered by any known treatment. You cannot alter your fingerprint no matter how hard you try, right? DNA
Yes, sir.
fingerprinting is now used for what? Forensic laboratories for identification of criminals. You can identify a criminal if you have its fingerprint record, right? To determine paternity of an individual. Like if you are someone's son or like if you are related to someone, you can have an idea with the help of fingerprint as well. The what is the next point? To identify the dead bodies in any accident by comparing the DNAs of parent or child, right? And the last one is to identify racial groups to rewrite biological evolution. Is there any problem about this DNA fingerprinting? It's just normal
No, sir.
fingerprint stuff.
Yes, sir.
Now, this is a bit important. From here, there are frequent questions asked in board exams, right? So basically, you need to be aware of two types of vitamins. First type of vitamins are those which are fat soluble, and the rest are water soluble, right? So how I remember it was like that. You know what is Kida? Kida is an insect, right? In Urdu,
Yes,
we call
sir.
Kida an insect. So these Kida vitamins are fat soluble, and the rest are water soluble, right? About
Yes, sir.
vitamins, you just need to be aware of this. And there are some few more points about these. These are stored in liver and adipose. Adipose is a fat storing tissue, right? Just a few more detail about it. These are few examples of water soluble vitamins. You don't need to learn them. Just go through this, right? You don't have to learn this. The only thing that you need to remember is water soluble fats must be supplied regularly in diet because they are readily exerted in urine, right? The only vitamin, this one, cannot be stored in our body. The vitamin B12 is the only vitamin which is water soluble and can be stored in our body, right? All the rest
Yes,
of
sir.
water soluble vitamins will be excreted through urine, right? These are like this is the vitamin and this is the source of the vitamin, and these are the diseases which will a person like have if they are these like if someone is vitamin A deficient, then he will observe this disease where hardening of cornea of eyes or night blindness, basically eye related issues if you are having the deficiency of vitamin A, right? If you are having the deficiency of vitamin B, then like you can see the loss of appetite, right? And these are some sources. Again, you don't have to learn this, just go through, right? The important thing is what are fat soluble vitamins? This KIDA and the rest are water soluble. This is the most important thing that you should remember.
Yes, sir.
Now, enzymes. Do you know what are enzymes? Any idea? Any guess you would like to make?
No, sir.
Right. You have heard the term catalyst.
Yes, sir.
What does a catalyst do to a reaction?
Sir, it increases the speed of the reaction,
Good,
how
good,
much
good.
time it takes.
Good. So what is enzyme? Enzyme is basically our biochemical catalyst. In our body, the thing which act as a catalyst are enzymes. It is just a catalyst, but in biological system. Is it making sense? Inside your
Yes, sir.
body, the thing which is like doing the work of a catalyst is called enzymes, right? So
Yes, sir.
first of all, it is enzyme. So it is a catalyst. Catalyzes the reaction in the body. It increases the rate of the reaction inside our body. A small quantity can do millions of the reaction. The best catalyst is enzymes, which is produced inside living organisms. Right? Now, this is a simple mechanism of enzyme. We also call it lock and key method. So if you try to relate it with this lock and key thing, suppose you have a key. Your key is substrate, and you are fitting that key into an enzyme. Suppose you can understand it. I will tell you another example as well. So first of all, let us go through this methodology. You have a key. Your key is what?
Substrate.
What is a substrate? Substrate is a thing where the attacking reagents are attacking and making the product. Suppose, if do you have any copy? Suppose you have a copy, right?
Yes, sir.
And what else you do? You have an enzyme. So enzyme. Okay, we will go one by one. First of all, let us understand what is this lock and key mechanism. Then I have a very good example that I will put, that I will tell you. So this key is substrate and the lock is enzyme. The enzyme is doing what? Enzyme, the lock is holding that key inside it, right?
Yes, sir.
Then we will open the lock. The moment the lock is open, we will get our product. If we try to understand it with this diagram, we will have a better understanding. This is our substrate. This is our enzyme, right?
Yes,
Enzyme
sir.
is providing what? The surface. Enzyme is providing a site where the substrate can fit into, right?
Yes, sir. Okay.
After the enzyme and substrate are together, they are making a complex. Once the substrate is at the surface of enzyme, it will start doing reaction. After some time, they will break and form product. The enzyme is just providing a surface where the reaction is happening and the substrate is broken into products, right?
Yes, sir.
You have a substrate, you have an enzyme. Uh, I think I did it. Just give me a second. This is your enzyme, this is your substrate. Enzyme is doing what? Enzyme is providing a site where a substrate can rest or fit into. And the moment they are together, there is a start of reaction in the substrate. And after some time, the substrate will break and give us product. And the enzyme will remain unaffected. You can understand it with another example. Hmm. Suppose you have to go somewhere in a car, right? You have to go
So,
somewhere in a car, and the thing is, you need to change your clothes. You are going in some party, but you are very late. So you are thinking you will change your clothes in car. So the starting you are substrate. The moment you sit into the car is working as enzyme. Car is providing you a place where you can do a reaction. You can change your clothes, and car is taking you from one place to another, right? The moment
yes,
you
sir.
change your clothes and reach your destination, you are converted from substrate to what? Product. You are ready to attend your event. Is
Yes,
there
sir.
any effect on car? Car can do
No,
this for
sir.
this like several times. Uh, right. Car is having no effect. It is just providing a surface from where a substrate can go from one place to another, and the substrate can do reaction and give us product. Is this clear?
Yes, sir.
A simple example of the enzyme reaction is this. You have a maltose, which is a disaccharide. What is a disaccharide? A carbohydrate, a protein, or nucleic acid. Question for you. What is a monosaccharide? You must know this. This is the first thing.
Cannot be it cannot be broken down
Okay,
into
what is it?
simpler
Carbohydrate or protein or nucleic acid? Which category does saccharides belongs to?
carbohydrate.
Carbohydrate, right? So this maltose is a disaccharide, basically a carbohydrate. And this maltase is what? An enzyme. This carbohydrate will sit onto the surface of this enzyme and go from one place to another. There will be some reaction occurring in this maltose, and after some time, the enzyme will give product glucose, 2 units of glucose, right? This is a reaction
Yes,
of
sir.
enzyme. You can see some other examples as well. If you want to make ammonia plus CO2, the substrate is going to be urea and the enzyme is going to be uranase. The thing which we discussed, maltose, the enzyme is maltase and the product is glucose. So if you do like one or two examples, they are sufficient. You can
Sir, so
do
sir, in the reaction we'll get in the reaction of maltose and maltase we'll get glucose and maltase, right?
no, no. Maltase. The enzyme will remain unreacted and will be left in the reaction. Like, yeah, you are correct. We'll get maltase, and this maltase will again absorb some more maltose and do the same process again and again and again.
Yes.
Right? This is
Yes.
just the car. The thing, the main task of it is to deliver passenger from one place to another. Is it clear?
Sir.
Now, some theory about hormones, and that's it. What are hormones? Hormones are molecules that act as an intercellular messengers, right? Have you
Yes,
like
sir.
I in 10th you must have biology. Do you have biology in 11th or not? Like, did you have biology
No,
or
sir.
are you a
Sir,
PCM
in
person?
sir,
Physics,
PCM.
chemistry, maths. Okay.
Yes.
Right. So you just have to treat it as theory. You just have to learn it. You don't have to go into the details because that will involve biology. So what are hormones? You can say hormones are molecules that act as intercellular messenger. These are produced by endocrine glands in the body and powered directly in the bloodstream, which transports them to the site of the action. Right? Some of these are steroids, example estrogens and androgens. Some are polypeptides, for example insulin and endorphins, and some others are amino acids derivatives such as epinephrine and norepinephrine. Right? Hormones have several functions in the body. They help to maintain balance of biological activity in the body. Right? See, in my experience, they haven't asked much from this hormone thing. If they will be asking anything, it will be a short type of note. So you can remember 4 or 5 points and you can write it, right?
Yes, sir.
So this is about the chapter. We have missed few topics from carbohydrates. Let us talk about them. See, I'm just giving you a brief again. Or let me say, I have added few more things. Like I was checking the previous board exam now, so there were some questions. So the thing we have studied, but they have just modified it. So I have wrote it down so you just don't get confused. A question was like, let me see about the preparation stuff. Yeah, the question was if we are doing the hydrolysis of sucrose, we are getting glucose and fructose, right? This is the preparation part. Do you
Yes, sir.
remember this or not?
Yes, sir.
If we do the hydrolysis of sucrose in acidic medium, basically we are boiling sucrose while adding some dilute HCl or H2SO4. So we are getting glucose and fructose. The question was the mixture that we are observing, the mixture that we are getting, is it levorotatory or dextrorotatory? Right? So do you know what is levorotatory and dextrorotatory? I have discussed it
Yes,
while
sir.
we were talking about. So tell me what is levorotatory and dextrorotatory?
Sir, in liver rotatory, the plane polarized light uh, will fall on the
Alpha
left
or
side,
left?
and for dextrorotatory on the right side.
Good. So the mixture is, see, first of all, the naming is like this. This D is signifying what? This D is just relative configuration, relative to the isomer of glyceraldehyde, right? This
Yes,
DE
sir.
relative configuration. But this plus or minus sign is absolute configuration. It is defining the dextro or levo term. So we are getting a mixture of dextro and levorotatory, right? The concentration
Yes,
is
sir.
50-50. But the optical activity of fructose is higher. The optical activity of fructose is higher. It is around 100 to 102 than glucose, which is 52. Thus, the solution of fructose and glucose, even though they are present 50-50%, mixture is overall levorotatory. Is it clear? Why? Because the optical activity of fructose is higher, right? So the mixture
Yes,
is
sir.
overall levorotatory. The rest is simple. Maltose, if you are doing the hydrolysis of maltose, you will get 2 units of glucose. Again, you are just boiling it in acidic medium, and you can do the same thing from starch. With starch, you will get number of glucose units, so many units. That's why it is used as commercially to produce large amount of glucose. Right. Then we talked about the how the structure is straight line, right? Chain structure. Then why it should be ring type because it was not showing some specific reaction. That's why we thought we treated it like a ring type of structure. Then it explains of this is what Harvard production, right? And if we are drawing this OH at the below side, it is alpha. If it is at the top side, it is
Beta.
the important part. The anomeric carbon is which carbon? The carbon number one is anomeric carbon, right?
Yes, sir.
They might ask you what are anomeric carbon. So you have to do what? You have to say in the ring structure of glucose or fructose, the carbon number one for glucose is our anomeric carbon. The position of the OH group on this carbon will decide whether it is going to be alpha or beta. So you can say alpha beta forms are called anomers and the C1 carbon is anomeric carbon, right?
Sir, so every time the C1 carbon will be the anomeric carbon, right?
For glucose, it is going to be always the case. Like the for glucose, the C1 carbon is always going to be the anomeric carbon. But for fructose, it is going to be the C2 carbon.
Okay, sir.
Anomers are diastereomers. Diastereomers are basically suppose they are optically active compounds, but the mirror image are not superimposing. For example, mm, okay, mhm. How should I explain this? You are saying the carbon number one, right?
Yes, sir.
The only different thing about these two structures is what? The position of carbon
The
number one's OH group, right?
Yes, sir.
So you can call them diastereomers. This is a topic of stereochemistry, then I have to go in a bit of like it will take so much time. So for now you can understand it. Anomers are diastereomers, right? They won't ask you much about it. They will ask you just this term. All anions are diastereomers, but all diastereomers are not anomers, right? If we are having several anomers, they are always going to be diastereomers. Whenever you are hearing the term anomers, then you can say they are diastereomers. Whenever you are hearing the term anomers, you will say they are diastereomers. It is always the case. But all diastereomers are not anomers. They might ask you in assertion and reasoning this type of questions, right? So
Yes, sir.
keep this statement in mind. What is mutarotation? Equilibrium between alpha and beta form is called mutarotation. This is our alpha form, right? Alpha form of cyclic glucose. This is beta form of cyclic glucose. If they are in equilibrium, we will call this process what? Mutarotation. The alpha and beta term in equilibrium is called mutarotation. Is it clear?
Yes, sir.
Now what else? See here, the C2 carbon is anomeric carbon. Why? Because the attack was done from where to where? From this fifth carbon to second carbon. The carbon getting attacked is the anomeric carbon. If you look over here, the carbon which is getting attacked is which one? Second carbon. So hence it is
Yes, sir.
anomeric carbon. In the case of glucose, the attack was done on the first carbon. So there the anomeric carbon was first, C1. Right. Now what
Yes,
else
sir.
to discuss? Yes. These are some different types of monosaccharides. We have talked about it, right? If we are talking aldose, the main chain is going to be 3 carbon consisting of 3
Yes, sir.
carbon. If we are using tetrose, it is going to be 4 carbon. So now we are going to talk about some specific example of monosaccharides. I will tell you which monosaccharides you need to remember and which you not. So first of all, this is what? Diglyceraldehyde. You should know because this is the reference one. With the help of diglyceraldehyde, we do what? We give relative configuration whether it is going to be D or L, right? If our compound resembles this one, here the OH group is at the right side, we will call it D. If somehow it is resembling a compound of glyceraldehyde where the OH group is at the left side, we will call it what?
Triglycerides.
Right? Now, aldotetrose. For aldotetrose, these two are important. First one is
Sir,
erythrose. Erythrose is
sir,
the compound consisting of 4 carbon. Here, the OH group will be at the same side. If we have a aldotetrose, a carbohydrate consisting of 4 carbon, and both of the OH group are at the same side, that we will call it erythrose. You
sir,
have a question?
sir, in diglyceraldehyde, uh, we don't put like positive or negative,
For diglyceraldehyde,
like D,
no, I
then
don't.
positive.
It would have a optically activity, like it would either rotate the right to the left or right. But for that, we need to check the experimental data. Let me see if I have written the experimental data set here. Yes, it is plus. It will rotate the light to the right hand side. It is optically active and it will rotate the light to the right hand side. It is dextrorotatory. Is it clear?
Sir,
But it is not necessary that if we are talking about D configuration, if we are talking about D configuration, the optically activity is always going to be dextrorotatory. It is purely a experimental phenomena. For example, see, if you look at the fructose, in fructose, here the relative configuration is D because at the last asymmetric carbon, the OH group is at the right side, but it rotates the light to the left hand side. Hence, we are using the minus sign. So, relative configuration and absolute configuration has no direct relationship. Relative configuration just relate to the glyceraldehyde isomer, but the absolute configuration is purely experimental. You have to pass the plain polarized light through your compound, and then you will decide whether it is dextro or levo. Is it clear?
sir.
Relative and absolute configuration has no direct relationship. Now, this is erythrose. Erythrose is what? It is aldotetrose. And what is special about erythrose? Both of the OH group are at the same side. Similarly, we have triose. In triose, everything is same, but the OH group are at
Other side.
opposite side. You have to remember this. The way you remember the glucose structure, you have to do the same for this. Now we will talk about some carbohydrate where the main carbon chain is of 5 carbons, right? So we will call them aldopentose. So here again, all of them are important. If all the OH group are at the right side, we will call it ribose. If the first OH group is the left is at the left hand side, we will call it arabinose. If we have a combination similar to this OH, 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 we will call it xylose. And if we have a combination like this, wait, just give me a second. This structure is not complete. Hmm, is xylose. Right. If we are having a combination like this, we will call it lyxose. Okay.
This way.
How will they ask the question? They will say, uh, they will ask in terms of epimers. I will tell you about what are epimers. So they will tell you, uh, they will ask you ribose and arabinose are what type of epimers, right? So for that we need to discuss epimers first. So give me a few minutes, then we'll talk about epimers. So we have 4 types of carbohydrates consisting of 5 carbon main chain. Now for aldohexose, you just need to learn 3: glucose, mannose, and this galactose. The most important aldohexose are glucose, mannose, and galactose, right? We will talk about all of them, but you just need to remember these 3. If you have time, you can remember all of them, but you should not leave at least these 3. These 3 are most important: glucose, mannose, and galactose. So if we are talking about aldohexose, first of all, the name is suggesting we are talking about a thing. Hex means 6 carbon, right? So if all the OH group are at the same side, or we can say all the H group are at the same side, then we will call it allose. If we have a combination like this, we will call it altrose. Then you are aware of glucose. This mannose is what? For mannose, what we are doing, it is similar to glucose, but at carbon number what? At carbon number 2, the OH is changed. In the glucose, at carbon number 2, OH is at the left right side, but for mannose, it is at the left side. The rest is what?
Same.
Same. Right. This is what glucose, this is iodose, and this is galactose. For galactose, again, you have to relate it with glucose. They will ask you question in relation with glucose only. In glucose and galactose, what we are observing? If you look at the carbon number 4 and carbon number 4, in glucose and galactose, what is different? The position of
OH.
OH. So now we will talk about epimers, which is very important. Again, similar to anomers, epimers are also diastereomers, right? Change in configuration at any one carbon is known as epimer. What? If at any carbon, we are changing the configuration. By configuration, I mean the position of the respective groups. If I am changing the respective group positions at any point, at any number of carbon, then I will call those pair epimers. For example, suppose here we have glucose and galactose, right? They will ask you glucose and galactose are which type of epimers. They will ask you glucose and galactose are which type of epimers. So first of all, you need to be aware of the structure of glucose and galactose, right? So by
Yes, sir.
now, I hope you know what how a glucose structure look like. At least you should know what is glucose, right?
Yes,
And
sir.
if we see, This is same. This is same. This is same. This is same. Right? This is same or no? This is different. This position is different, and the rest again same. So, as per the definition of epimer, what is epimers? Change in configuration, or we can say change in the position of any two groups at any one carbon is known as epimer. Here, at carbon number 4, the position of OH group is different in case of glucose and galactose, so we will call them epimers. And which type of epimers? Carbon number 4 epimer. Is this clear?
Yes, sir.
Now, similar to mannose. What is mannose? At
C2
carbon number 2,
epimers.
what we are observing? Change in configuration. So we will call this glucose and mannose are C2 epimers of each other.
Sir,
Is
but
it clear?
there was one more, right?
One more what?
One more C2 epimer of glucose.
One more C2 epimer of glucose. Okay, let me see. Which one?
Sir, oh, okay, that was mannose. Yes.
Yeah.
I thought that was something different.
These two are the same thing, right? We were talking about mannose
Yes,
here.
sir. Yes.
Is it clear? These two
Yes, sir.
questions again are very important. They will either ask you mannose and glucose relation or galactose or glucose relation. So you should be aware of the structure of both of them. You don't
Yes,
have
sir.
to like remember it. You just have to know the glucose structure. And if you are changing the position of OH at carbon number 2, you will get mannose. If you are changing the position of OH at carbon number 4, you will get galactose. So what you need to learn?
Sir,
Just the structure of glucose. Yeah.
sir, they will ask mainly only these two, right?
These are the most important.
And epimers. Okay, sir.
These are the most important ones. If you see the previous year questions, they haven't asked apart from these two till now.
Yes, sir.
Now we will talk about disaccharides a bit. What are disaccharides? When two monosaccharide units combine together. Simple. So if I need to make maltose, here we will clear a very important concept, reducing and non-reducing sugars, right? So please focus carefully. So first of all, if we are talking about disaccharides, we know we have to use two monosaccharide units. A glucose is a monosaccharide. So I am using what? Two alpha D glucose units. Here I should have wrote glucopyranose, right? Because the ring structure of glucose is said like written as glucopyranose, but you can use this short term as well. In your exam, if they have given you alpha D glucose, so you just have to assume if they are using alpha or beta form, they are talking about ring structure, right? If they are not writing the whole thing, glucopyranose, and just mentioning the alpha part, so you should yourself understand it as ring structure, right?
Yes, sir.
So we are using what? Two alpha D glucose. The if we are talking about alpha, we know the OH is going to be at the downside. So we have to connect these two units, right? So how we are going to do it? Basically, whenever we are forming the maltose, we will observe condensation. By condensation, I mean we will observe a like we will basically see a loss of H2 water molecule. So how we are going to observe it? Try to look over here, right? If I am doing this, what is getting removed? H2O.
Sir.
And if I am removing this H2O like this, then what I will observe? I will see a bond like this or not.
Yes, sir.
Let us do it again. The H2O will be removed and this bond will look like this. Now, we have been talking about glycosidic linkage, right? So we will now see how a glycosidic linkage actually looks like. So here, after the removal of H2O, this these bonds will be connected in this way, right?
Yes, sir.
Or not? This bond is what? Our glycosidic bond. And what we are observing here, the bonding is occurring at which position? Here the carbon number 1 and this carbon number 4 are getting connected, right? The linkage is
Yes, sir.
happening at which number of carbon? Carbon number 4 and carbon number 1. So we will call it which type of glycosidic linkage? Alpha 1,4 glycosidic linkage, right?
Yes,
And
sir.
how our maltose is going to look like? Our maltose is going to look like this. What we did? We just took two alpha D glucose unit and we connected it with carbon number 1 and carbon number 4 by eliminating H2O. And the thing which we are getting after doing this is our disaccharide, which is specifically maltose. And maltose is a reducing sugar. Now we will try to understand how we are saying it or how we are categorizing it into a reducing sugar. Are you looking at this anomeric carbon? This is anomeric carbon, right? The carbon number 1 is anomeric carbon.
Yes, sir.
Here at anomeric carbon, we are having free OH group. Here the OH group is available, right? It is not participating in any type of bonding. It is freely available, right?
Yes, sir.
So in disaccharides, where at anomeric carbon, the OH group, the hydroxyl group is free, those carbohydrate will be termed as reducing sugars. Or you can say this hemiacetal term is used in alcohol chapter. So while we will talking about like in alcohol, while we will discussing this term now, I will explain you in detail. So for now, you can understand it this way. If we are having a group like this, we'll call it hemiacetal, where we are having one OH and one OR group. If we are having two OR group, then we will call it acetal. Right? For now, so that you can easily understand, you can relate it in terms of anomeric carbon. If at anomeric position the OH group is free, those sugars are what?
Reducing sugars.
Reducing sugar. Anomeric OH group. Right? And the bonding is alpha 1,4 glycosidic linkage. Similarly, we will talk about lactose. For lactose, we are using beta D galactose and beta D glucose. By beta, what do I know? The OH group is at the
Top.
top side. Now, you must be wondering how we are going to form the cyclic structure of galactose. We haven't talked about it, right? So how we are going to make the structure of it, right? So you just have to do simple thing. In galactose and in glucose, what is different? At carbon number 4, the position of OH, right?
Yes, sir.
So this is the structure of what? This is the structure of glucose. This is the structure of glucose, beta glucose. If if at carbon number 4, at carbon number 4, I am replacing this OH from bottom to top, then I will call it what? The cyclic structure of galactose. Just by
Yes, sir.
just by replacing or changing the configuration of carbon number 4 OH group, I have converted glucose into galactose. This is beta D galactose and this is
Beta D glucose.
clear. Again,
Yes,
what is
sir.
going to happen here? They will condense with each other by eliminating one H2O unit, and they are doing this process from carbon number 1 to carbon number 4, right? So
Yes,
again,
sir.
we will observe something like this. From carbon number 1, see, You must be wondering one thing. Here we have made this bond in the like this, right? In the above trend. But in the case of maltose, we did it slightly downhill type, right? Why is this? So if you see here both of the OH group will were at the bottom and side, right? So if I
Yes,
have to
sir.
connect them, if I have to connect them, I have to use this downhill type of bond. But if I am using this and I have to connect this, then I have to do this or not. I have to, if I have to eliminate water molecule, I have to take this beta D glucose unit a bit slightly above so that I can
Yes,
connect
sir.
it and I can eliminate it.
Yes, sir.
That's why we draw lactose like this and mannose like this, right?
Yes, sir.
Again, everything is same. You will connect this and you will call it beta 1,4 glycosidic linkage. Why? Because the OH group was beta. Is it clear?
Yes, sir.
Now here, if it see this carbon number 1, this anomeric carbon, the OH group is free.
Yes, sir.
So it is again what?
Reducing sugar.
Reducing sugar. Now, the structure of sucrose. Here you will see this anomeric carbon, which we were observing as free till now. We will see that OH group participating in bond. For sucrose, what we need? Alpha D glucose and alpha D fructose, right?
Sir.
So this is our alpha D glucose and this is fructose. What is happening here? Here, the bonding will occur from carbon number 1 to carbon number 2. Again, they will eliminate one H2O unit and they will connect in a manner like this. Here you will see the anomeric carbon position is this. Carbon number 1 is anomeric carbon position for alcohol, oh sorry, glucose. And,
Sir.
and, and for fructose, the anomeric position is what? Carbon number 2.
Yes, sir.
At carbon number 2 and carbon number 1, are you observing any free OH group?
No, sir.
See, let me do it. Suppose we have these, right? I am doing what? I have to make the glycosidic linkage from carbon number 1 to carbon number.
2.
So the OH group that I'm going to use are these two, right? What
Yes,
is
sir.
going to happen? This H will be eliminated with OH minus H2O, right?
Yes, sir.
Then I will see what? I will see these bond connected in a way like this. This O will remain here and we will see this O like this, right?
Yes, sir.
Now, this position for glucose, the carbon number 1 is anomeric carbon. And if this OH group was free, then I will call it reducing sugar. But here I am observing both of the anomeric carbon for fructose as well as glucose. The OH group is not free. They are bonded with each other. They are bonded with each other, right? So this type of sugars are
Reducing
non-reducing.
sugar.
Is it clear now what are reducing and non-reducing sugar for disaccharides?
Yes, sir.
Every monosaccharide is reducing sugar. But when we talk about disaccharide unit, we will see the anomeric carbon. If the anomeric carbon OH group is free, then it is reducing. If not, then it is non-reducing. Okay.
Yes, sir.
Now, some few points about polysaccharide because in last exam they asked difference between cellulose and glucose, right? So let us talk about polysaccharide as well. What are polysaccharides?
There are
More than 10
many,
units of
yeah, more than 10 units
carbohydrate
of monosaccharide.
monosaccharides are joined together. Or we can say several units of monosaccharides joined together is called polysaccharide. Poly means several, right?
sir.
So what is the purpose of polysaccharide in nature? Polysaccharides serve mainly as food storage and structure material in nature, right? The important thing is starch. So what is starch? Starch is the main storage polysaccharide in plants and a major dietary source for humans. We use starch in what? If you like, you eat rotis, right? Rotis are made of wheat and wheat contain what? Starch. So we are consuming starch. So it is a major dietary source of for humans. Okay, because it is present in wheat.
Yes, sir.
It is found in cereals, roots, tubers, and some vegetables as well. Starch is polymer of alpha D glucose because if you are breaking starch by boiling it in water, we talked it about in commercial method of preparing glucose. If you are breaking a starch in it, you will get several number of glucose in it. That's why it is used to produce glucose commercially, right? So which type of glucose it produce? Alpha D glucose, and it has two components: amylose and amylopectin. What is amylose? It is water soluble. You don't have to write this, right? You just have to write what? Water soluble and water insoluble. Okay. This
Yes, sir.
is unbranched and this is branched. This is formed by linkage of C1 and C4. This have C1 and C4 glycosidic linkage similar to mannose and lactose. But here we have C1 C4 linkage in the chain as well as C1 C6 linkage at the branches. Right. These are few points you can add if somebody is asking you about amylose and amylopectin. How they look like? They look like this. You don't have to remember the structure and they won't ask you to draw the structure either. You just have to look it to understand. You don't have to learn it. This is so that you can understand how the linkage is formed. For amylose, we have 1,4 glycosidic linkage only. But for amylopectin, we have 1,4 glycosidic linkage. This is 1,4 glycosidic linkage plus 1,6 glycosidic linkage as well. This is the reason they are branched. And due to the absence of it in amylose, they are chained. Right. So
Yes, sir.
just take few minutes because I think it is getting a bit overwhelming, or is it easy? Am I going too fast?
No, sir.
Sure.
Yes, sir.
If at any point you are thinking we are going a bit fast, you can tell me. I will slow. Now, what is cellulose? Again, it is a carbohydrate, right? And one more thing. The molecular formula of the molecular formula of starch and cellulose is same. If we talk about the molecular formula of glucose, oh sorry, can I stop? Starch and cellulose. The molecular formula of starch. The molecular formula of starch is this, and it is same for our cellulose as well. But their stereochemistry is different. Their stereochemistry is different. We will talk about it. Just give it. Let us talk about what are cellulose first. Cellulose is only found in plants, and it is most abundant organic substance in plant kingdom. Its main component, it is the main component of plant cell walls. Cellulose has straight chain polysaccharide made up of beta D glucose. The only difference in starch and cellulose is what? In cellulose, we have alpha glycosidic linkage. In straight chain, we have alpha link, right? In beta,
Yes,
we
sir.
have beta. Sorry, in cellulose, we have beta linkage. Rest is same. Again, C1 to C4. This structure will look like this. Why we have drawn it like this? Because if we are connecting to beta OH group, we have to align them in this, like this, in similar way of lactose. Because if we have to connect two beta units, we have to align them in a stacking method, in a stay type of thing, right?
Yes, sir.
This has a very important significance just because of the different orientation. Just because of the different orientation, just give me a second. See, in starch, the linkage is straight, right? Like this. Just by changing the position of the two units, just by changing the position or changing the orientation of the glycosidic bond from alpha to beta, this has very like significant uh, consequence here. Due to this orientation only, due to this different orientation only, we are able to digest starch and we are not able to digest cellulose. Just due to the stereochemistry, just due to they are differently orientated in space, we are able to digest starch but not cellulose. In starch and cellulose, they have same molecular formula, they have same number of atoms, but the orientation is different. That's why we are not able to digest cellulose. On the other hand, we are able to digest starch. Is it clear?
Yes, sir.
Now, what is glycogen? Glycogen is storage from form of carbohydrate in animal, also called animal starch. Okay, structure is similar to amylopectin, but more highly branched, found in liver, muscle, and brain when glucose is needed. Enzyme fail. This is its definition, right? Try to read it out. If there is any point that you are not able to understand, ask me. Just give it a read. This is the last topic of this chapter.
Sir.
Is there any point or any term that you are not understanding? You can ask.
The fourth point.
Suppose glycogen are present inside the body, right? Inside our body, right? So if
Yes,
somehow
sir.
you are having shortage of glucose, if you are having shortage of glucose, inside our body we have so many enzymes. An enzyme is basically a catalyst. Suppose if your body is in deficiency of glucose, so they will convert this glycogen by breaking it into glucose, and your body will be able to get glucose, right? So if your body is deficient of glucose, they will convert the glycogen into glucose by the enzyme mechanism. That's it.
Yes, sir.
Suppose in summer, we have deficiency of glucose now because we are sweating too much, right?
Yes, sir.
So at the moment where your body has no glucose left, the glycogen will be converted into glucose. You have to survive somehow. So your survival instinct will kick in and the glycogen will be converted into glucose.
Yes, sir.
And if this glycogen is exhausted as well, then you will tata bye bye. Now, a question here. What is glycogen and how it is different from starch? So you can write a few points about it. Glycogen is carbohydrate, polysaccharide. It is present in animal. Okay. Glycogen consists of only one component, whose structure is similar to amylopectin. Also, glycogen is more branched than amylopectin. What is the difference? It is more branched than
Branched.
amylopectin,
There are multiple.
right? What is its use? It is used to in animal carbohydrates are stored glycogen as glycogen,
Yes, sir.
right? So just give me one more minute. Meanwhile, this is the I think they have asked this in the previous board. So they asked what is the difference between starch and cellulose. So try to read these points, then I will explain. Just give it a brief read. You will be able to understand it because we have already discussed all the points that are written in. Just give it a read for a few minutes. So first of all, occurrence, where we are finding the starch and where we are finding the cellulose. Starch is found in plant as a storage carbohydrate, and cellulose are found in plant but as a structural component. Okay,
Yes,
they
sir.
are both found in plants, but their uh, what do you say, properties are different. One is used as
Question.
storage of carbohydrate, the other one is used as structural component. Their function, their function are what? Energy storage in plants and humans.
Yes, sir.
Starch is used to store energy in plants and humans, but cellulose is used to provide rigidity to plant cell wall. If we are talk about monomer unit, here we have alpha unit, here we have beta unit. That's the reason of the different orientation of, right? They have different orientation because of these alpha and beta units.
Yes,
If we
sir.
talk about structure, they have mixture of amylose and amylopectin. What is amylose? Amylose is unbranched and amylopectin is branched, and they have only unbranched or straight chain. They don't have branch type of unit. The
Yes, sir.
linkage is alpha 14 and alpha 16. Here we have only beta 14. Digestibility. Starch is digestible by humans and cellulose is indigestible by humans. That's why cellulose is present in grass. The grass which is eaten by cows and goats, right? Cellulose is present in grass. And animals like cows and goats are able to digest that grass because they have enzymes in their body which are able to digest that. But for humans, we don't possess that enzyme, so we are not able to digest cellulose. On the other hand, we are able to digest starch because in our body we have enzymes which are capable of breaking down starch. Is it making sense?
Yes, sir.
And why they are different? They have every single atom is same, right? Molecular formula is same, just due to the different orientation of the bonds in 3 dimension. They have entirely different property, right?
Yes, sir.
Solubility. Amylose is partially soluble. Amylopectin is insoluble, and this is insoluble in water. So you can write few points about it, I guess. Now,
Yes, sir.
if they ask you what is the difference between cellulose and starch, you can write few points about this. Right? Let me see. We talked about 20 different type of proteins, right?
Yes, sir.
20 different type of sorry, amino acid, not proteins. 20 different type of amino acids joined together in different sequences to produce different proteins. So if you need to see which type of amino acids do we have actually? So these are 20 different type of amino acids. The star one are which type? The star ones are essential amino acids and normal one are non-essential. So out of essential and non-essential, which are required to external diet?
Essential.
Good. So the star ones are essential amino acids that you need to take from diet. So how will they look like? First of all, how does a alpha amino acid look like? At alpha position, what we have? Amino group. And alpha position is respect to what? Carboxylic group. So this is the basic structure of or basic formula for amino acids. You have to just replace R. If you are replacing R with what? H. Suppose if I am writing this. If I have replaced R with H, then this amino acid, this alpha amino acid is which type of amino acid? This is called,
I see.
if I am replacing my R with what? CH3. Then it will be alanine. Similarly, if I'm replacing it with this CH, CH3 and CH3, then it will be valine, right? You don't have to learn all of them. I have given you a specific example of dipeptide, right? Where we were adding lysine with alanine. We were basically joining them by eliminating bottom molecule.
Yes, sir.
Let's see, this one. So if you try to remember at least two example of this, then they are sufficient, right? You can choose whichever alpha amino acid you are most comfortable with, you can learn, right? But these two basic one, you should always remember because these are very simple. You just have to replace R with H and CH3. The rest is same. So at least you should be aware of glycine and alanine. And a very frequent question is what? They will ask you which type of amino acid is optically inactive. Only one amino acid is optically inactive. Which is that? I've told you several times. Which type of amino
Glycine.
acid is glycine? Glycine. Good. And you should know the structure of glycine and alanine at least. The rest you can see from me. So that was all about the biomolecules chapter. Have you given the test or you are going to give it?
I give the test.
Okay, so shall we discuss the test? We have some time.
Yes, sir.
Okay, let us discuss the test. Give me a few minutes. In the next class, we will start the haloalkane chapter and try to revise the three chapter we have already done because you have your exam on 11 or 16. So if you delay, then we'll all just stack up and it will cause problem. So try to revise regularly for at least 1 hour.
Yes, sir.
You have to answer right. Is my screen visible?
No, sir.
Now it is
Yes,
visible.
sir. Yes.
So. What should be the answer of first one?
Primary structure.
The primary structure. Why? Because the sequence in which we are assigning the amino acids leads to the formation of the primary structure, right? This is
Yes, sir.
the hydrolysis of sucrose is called what?
Inversion.
Inversion. Okay. In this, proteins are polymers of alpha amino acids connected by peptide bond. Is this correct or not? In amino acids, in proteins, we use peptide bond or glycosidic linkage, or the next one is phosphodiester linkage. Which type of linkage is used in protein? Is this correct?
Yes, sir. Peptide bond.
Okay. A tetrapeptide contains 4 amino acids. Is it correct?
No, sir.
So the answer should be
C.
C. A is 2, but R is false. By
Sir, I wrote C.
you wrote C. Okay. C. The first statement is correct. Proteins are polymer of alpha amino acids connected by a peptide bond. A tetrapeptide contains 4 amino acids linked by a 4 peptide bond. Both of these statements are correct, right? But B is not the reason for A.
Yeah.
Proteins are polymer of alpha amino acids and they are connected by peptide bond. This is correct. A tetrapeptide contains 4 amino acids linked by peptide bond. We have already discussed this now. See. This is a peptide bond, right? Here,
Yes, sir.
how many amino acids we have used?
Four,
Try to see carefully. How
four,
many amino acid, how many alpha amino acids we have joined together? Glycine
two.
and alanine. Is there any other thing we are
Yes.
seeing? So we
No,
just
sir.
have added two units. So it will be a dipeptide. If you try to read this statement, carboxy group of glycine combines with amino group of alanine, we get dipeptide, right? The name of this amino acid is called glycyalanine. After joining glycine and alanine, we get glycyalanine. A dipeptide is formed. A dipeptide is formed by two amino acids. A tripeptide is formed by three amino acids, and a tetrapeptide will have four amino acids, right?
Yes,
A
sir.
penta will have five amino acids. So this is what we have discussed. So what should be the correct answer now?
B.
What?
B.
What is deuterion?
Sir, they contain negative and positive ions.
Okay, good. But which type of like is does it belong to protein, nucleic acid, or carbohydrate?
Sir, it belongs to proteins.
Proteins. And in protein,
Sir,
what is the basic
uh,
the monomeric unit?
sir, sir, amino acids.
And deuterion is for specially which type of amino acid?
Sir,
Alpha,
uh,
beta, gamma,
sir, alpha.
alpha amino acids, right? If we see
Yes,
the deuterion, it look like this. First of all, this is what if we have this to the next carbon of the carboxylic group, it is alpha carbon, and to this
yes, sir.
carbon we have amino group, so we will call it alpha amino acid. And if I am saying it to be a deuterion, then we will have both the charges in this species. Here we will have positive charge, and here we will have what?
Negative charge.
Negative charge, right?
Yes,
This is
sir.
what? Deuterion.
So,
So you should add few more points about the frequency. Where is deuterion? Okay, the signs are approved. This H, this will abstract this H positive, and these bond will be transferred to O. Then we will have like this, right?
yes, sir.
Because whenever we talk about acid's main purpose is to give H positive, and base main purpose is to abstract H positive. If NH2 is working as base, it will abstract that H positive and be a positive charge. And see, if it is understandable, then only say yes. Otherwise, ask me again. We have lone pair of nitrogen.
Yes, sir.
Lone pair of nitrogen will do what? They will try to pick this H positive, right?
Yes, sir.
If they are picking H positive alone, then the bond which has been formed by the contribution of two electrons, in single bond we have two electrons.
Yes, sir.
These two electrons will be left behind on
Yes,
carbon.
sir.
So carbon will bear which charge? Negative. Now, if this H is giving its own electron, its own lone pair, then it is donating its own lone pair, right? So it will bear positive charge. In organic chemistry, I used to like teach in a way like suppose if you are helping someone from your own money, then you will bear a positive charge, right? These lone pairs of nitrogen were its own, right? These lone pairs
Sir,
belong to nitrogen. So it is donating its own lone pair to help someone. Here it is helping what? H positive, then it will be a positive charge. And if we are having negative and positive charge in a species, we will call it deuterion. A deuterion is neutral or not?
yes, sir.
Because the negative and positive are equal.
Equal.
If question, if there are more number of carboxylic group than ammonia group, then it will be a acidic, basic or neutral ion.
It will have a negative charge, so it will be basic.
So just a question, are you like revising it from my notes or from some other source? Because if you are using some other source, let me share it from me first, right? I will see whether the definitions are correct or not.
No, sir, I'm studying from your notes.
If you are studying from the notes, it's great. Otherwise, if you are using any reference, please get it checked by me first. Otherwise, you will write something wrong in your exam. Okay. So let us discuss the other questions. The fifth one. What should be the answer? In double strand helix structure of DNA, the two strands are complementary to each other. Why?
They're complementary to each other,
Yes.
so
Why?
that
I
sir,
mark
like
this important. See, I told you na from double strand helix structure, they might ask you two nucleic acid chains are wound upon each other and held together by hydrogen bond. Doing this, the two strands are complementary to each other. Question mark. The answer is because the hydrogen bond is formed between specific pairs of bases, ATCG. Right. This
yes,
is
sir.
the reason. How does it look like? Like this. I told you, no.
Yes, sir.
The blue one is cytine, guanine, adenine, and thymine. These bases are connected in a way similar to this. The adenine is connected with thymine,
Thymine.
right? These are bases and they are connected in a specific order. This is the reason why they are complementary, right?
Yes, sir.
You should have been able to answer this one. This is very easy.
Yes, sir.
Fibrous and what?
Fibrous proteins are sir, they're like straight. They
Hmm.
are not and globular proteins are spherical shaped.
Okay. And if we talk about their solubility, which one is water soluble and which one is not?
Sir, fibrous is soluble and globular is non-soluble.
Okay. Have you written
Fibrous
some
are,
examples?
yes,
Uh,
sir, in keratin, and globular proteins are formed in hormones and enzymes.
Good, 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 good. See, these all are very frequent questions from this chapter. So if you are able to answer them, then you are on the right track. The next one. Pentaacetate of glucose does not react with hydroxylamine. Why?
I had not heard the answer.
You don't know the answer for
The
this?
first one. Yes, sir.
See, while we were discussing about the cyclic structure of what? Glucose, we observed the issue. While we had the pentaacetate of glucose, it was not further reacting with hydroxylamine. This was the reason which led to the concept or like idea that it there might be a case where the glucose is not in like state change structure, it has occupied a ring type of structure. So if we go back and see here, see, this is acetylation of acetylation of glucose. This is our pentacetate, right?
Yes, sir.
If I am reacting it further with hydroxylamine, I am not observing any reaction, right? But if I have free CHO group, if I have free CHO group, then it should have reacted. So this is what this is indicating. This is indicating that this CHO group is no longer available. So then we did what? We talked about the cyclic structure. Here you can see the pentaacetate of glucose does not react with hydroxylamine, indicating the absence of free CHO group. You should have wrote in hydroxylamine there is no CHO group. There is no free CHO group because the chain is converted into ring. The chain is converted into ring. That's why this reaction is not proceeding. Right?
Yes, sir.
Is it clear?
Yes, sir.
What about the second one?
Sir, amino acids behave like salts because uh, sir, because in their anhydrous form they behave like zwitter ions.
Hmm. Good,
So
good, good.
that's why they behave like salts
Okay.
because they
The next
contain
one.
both positive and negative.
The two strands in DNA are complementary to each other. This was a bonus question because this is same, right?
Yes, sir.
What are reducing and non-reducing sugar? Give examples.
The reducing sugars, uh, sir, in the sir, what was that?
Valine and tolerant reagent,
No, sir.
monosaccharide,
Sir, the first carbon.
monosaccharide,
No, sir. Sir, in the ring structure,
aldehyde
the
ketose,
first carbon name. No, sir.
uh,
For glucose in the first carbon and in fructose for the second carbon.
anomeric, anomeric,
Yes, sir. Sir, so in the anomeric carbon, if there is a free,
OH
uh,
group.
yeah, if there is a free OH group, then it will be reducing sugar. Or if there is no free OH group, then it will be non-reducing sugar.
But this is about disaccharide only, right? You are correct. You are absolutely correct. But this is the definition for disaccharides. But we have to talk about monosaccharide as well. So you should
Yes, sir.
start like this.
Sir, I wrote
All
like this only.
good, good, good. But the point you added is correct. In disaccharide terms, if the anomeric carbon having three OH group, then it is reducing, otherwise non-reducing. You can tell the
Yes,
example,
sir.
sucrose is non-reducing and maltose and lactose is reducing. All those carbohydrates which reduces Fehling and Tollens reagent. Suppose you have a carbohydrate inside your test tube and you are adding Fehling solution and your carbohydrate is turning into a brown color of fumes. It is producing brown color of fumes, right? Then it is reducing sugar. And
Yes, sir.
if you are adding Tollens reagent and there is a silver mirror at the surface of your tube, at the layer of the top layer of your like liquid of carbohydrate, you are having a silver mirror. Then it is again reducing. If they are
Yes,
giving
sir.
Fehling and Tollens, they are reducing. All monosaccharides, all monosaccharides, whether they contain aldose or ketose group, are reducing sugar. Our galactose, mannose, glucose, erythrose, triose, glyceraldehyde, all of them are monosaccharides and all of them are reducing sugars, right?
Yes, sir.
In disaccharide, you can add the point you already told me, right? If the anomeric carbon is free,
Yes, sir.
that's it. The last question is, what type of linkage is present in carbohydrate, proteins, and nucleic acid? You can answer.
In carbohydrates, glycosidic linkage.
Okay,
In proteins, polypeptide linkage,
just peptide
and in
linkage.
nucleic acids, phosphodiester
Phos-
linkage.
Great, great, great. So this was all about this chapter. From the next class, we will start our Hello again, right? And please
Yes,
try to
sir.
revise at least for an hour. Give it daily so that
Yes,
you
sir.
can have a like better grasp on the subject by the time you have your exam. So that's it for today. If you have any doubts from
No, sir.
previous chapter or this chapter, you can ask.
No, sir.
Okay, that's it for today then. Thank you.
Thank you.